And something that you're not used to me doing is I'm going to go ahead and share my screen right away. But just to everybody, as I'm getting this pulled up, thank you again for joining us for what is our last tutoring session for the 2025 competition cycle. And let me see where there we go. Okay, and we'll put this in the presentation mode. Could someone give me a thumbs up that you guys are seeing this in the right full screen view? There you go. Okay, perfect. Alrighty, so again, welcome. We're so glad to have you here. I'm gonna start kicking us off by going through a couple of things about your examinations that we have in just under 2 weeks. I know you're all really excited about them, and we've gotten a couple of emails asking a little bit more about what's going to be in these examinations. And so I wanted to put in here just a couple things, and let me just disable our waiting room really quick so that everybody can jump right in. There we go. Alrighty. And then after I get through this brief presentation, then we'll allow all of our 3 invited presenters to go through their content with you. So with that, we'll just jump right in and talk through where you could find all this. All this information that I'm talking about today is found on our website. You can find all of our learning objectives and what you're responsible for on our website. Just as a reminder, the local objectives are still fair game to be tested on, but we're going to put a priority towards these regional objectives that we added new to this regional competition. So you can expect a combination of maybe some of the content you've had before, but primarily a lot of this new stuff that's going to be found within these learning objectives. If you need any questions for what's on the exam, you can find all this content again on our website underneath the 2025 Regionals tab. Now, the written examination is going to be incredibly similar to what you saw in the local competition. It's going to be looking at the same lecture objectives, but now we have the regional ones within there. The examination is going to be 125 multiple choice questions, and you'll be given 2 hours to work through those questions. Again, there's going to be an emphasis on regional, but local objectives are fair game. We could have them be written or image-based, just as you had seen in the local competition. But a lot of the image-based ones that you're going to see too before, they could be moved to the practical. So so that you're aware that you could still see a combination of those despite the fact that we now do have a practical. Questions can be first or second order, and I have a quick slide to just run through what that means with you in a minute. And we are asking that you bring a personal device to the competition so that you can take the exam. If for any reason you don't have one or you need one to take the exam, please let your regional coordinator know as soon as possible. I know we put it on the registration, but if anything's changed in the meantime, if your personal device broke or something, then again, please just let us know that as soon as possible so we can make arrangements for you. We will not provide you with the subscores for this test until the completion of the competition. So you won't finish and get your score right away. You're going to have to wait until the conclusion of all of the competitions Sunday when we tally everything up to get your final results. Now, what did I mean by first and second order questions? First order question is just going to be a question where there's just one correct answer that is directly answering the one question that you have. So one example could be, what is the name of the bone in the arm? And so the only correct answer for that, the only bone that's found in the arm is the humerus. So it's just testing a very basic level of understanding with these first order questions. With the second order question, we're going to be asking a follow-up question that tests your understanding of the initial question we asked. So we could ask, what is the function of the tag structure, for example, like if we tagged the brachialis muscle, and then you would need to understand the function of that muscle. So not just knowing that it was brachialis, you go into the second order on top of that, of then understanding that it is in fact a flexor then of the arm. So these second order questions are a little bit more challenging, and you can expect to see a mix of first and second order questions on your examination. The practical exam is going to be found in our donor labs. So you'll be provided with all of the personal protective equipment that you need. You'll be provided with gloves, aprons, all those things before you even meet the donors. And you'll be provided with any materials you need for the exam. So all you need to do is show up with yourself and all of your knowledge for anatomy. The practical contains 50 multiple choice questions. 40 of these questions are going to be anatomy focused and 10 histology focused. And this will include tagged structures on donors, on real donors, on models. This could be on a whole or a disarticulated skeleton, and then also histological images that could be found either on a computer screen or printed. You are not allowed to touch the donor during the practical. That's just to make sure that everything is nice and clean and out there for you, and that whoever went before you doesn't accidentally mess up the tag when they're running through. So it's actually in your best interest that there will be no touching during the exam. Everything will be as clear as day for you to know what is being tagged. You'll have 1 minute per question, a total of 50 minutes then for this subtest. And the tag structures are only going to be limited to the ones found, again, within our tag structure list, also found on our learning objectives subpage on our website. We highly recommend that you use the atlases that our medical students have compiled together. Students from Drexel University and Thomas Jefferson University did such a great job in identifying and labeling these images for you, because we know that it's a little bit of a transition between finding them in a drawn image versus 3D. So please go through look through those atlases before you move forward with studying for the practical examination. Same as with the written, you will not be given any of the scores for this until the end of the competition.
Dr. Briscoe,
Now,
can I just interrupt just for a minute?
yeah.
Let's just mention that there's a short little video on how to, in fact, kind of prepare for your first lab practical, how you should kind of think through that. It's a YouTube video, and I don't think it's longer than about 10 minutes.
Yes, and that was also put together by students from Drexel University that did a really great job. And just as students had also gone through that transition of finding, how do you study versus on the computer to preparing for your in-person examination. And they did a good job of creating a YouTube video that is found on our website. And I'm going to go ahead and link it to this video once I post this one onto our YouTube channel too. So you guys can find that one pretty easily. Now, it should also be on our 2025 regionals competition tab underneath the practical information as well. So that should be accessible currently on our YouTube channel and on our website. Now, if you're worried about going into the donor lab and also then being in a testing environment for that, we are aware that this could be a sensitive environment for you, but compared to what we had for the overall excitement in the lab in the previous year with all of our, again, high school age students, we found that it was an incredibly rewarding experience for all to be there. So we will do a lab introduction for you to introduce you to the opportunity and what it actually took to donate your body so that you could have this opportunity. You'll get a chance to meet the donors ahead of time and spend a little bit of time in the anatomy lab asking questions so that you're not just thrown into the testing environment. So we'll make sure that you're well and acclimated before you actually have to do any testing specifically for the practical. Finally, we'll conclude with some reflections, and this will be done at the end of the competition on Sunday, just so that you're able to come to some conclusion and a really nice ending, essentially, to your experience there in the donor lab. And I think that'll really help you guys moving forward. Again, last year it went so well with our reflections that we saw. The students had a great experience being able to go in the lab, and overall, they were able to acclimate very well and really do great work on the practical. It was really exciting to see. Now, the next one is going to be a standardized patient exam, and I'm going to finish up with the oral exam. Now, when we're going through these last two, I do have one example of each to run through with you, just so that you have a little bit more experience for what this is going to look like. There are also an example of each posted on our website that you can work through. For the standardized patient exam, this is going to be a standardized patient is someone who is trained to portray these symptoms of a disease or an illness, and then provide some training then for a medical student or any other person in training to be able to become aware of what these symptoms look like and make a proper diagnosis. This is a really low stakes environment for students to make these diagnoses before they're actually doing so in front of a patient where their health is really depending on it. So these will be nice practice ways to test for the health of an individual. You will get to experience 10 of these at the Anato-Bee, and each one is going to be 5 minutes in length and identical across no matter where you're attending at the Anato-Bee, whether it's the Northwest or the Southeast. You'll be randomly assigned to work with another person at the competition. When you go into each standardized patient room or station, that standardized patient is going to read an illness script that characterizes a disease, and it could include any patient demographics that are relevant to the history of that patient and how that might help you better form a diagnosis, any history of the symptoms that they're having, and then a time course, whether this is something that's been ongoing or something more recent. Now, each student is able to ask 2 questions from a list that's provided online. Again, that's something linked on our website, and I'll put these up for you so you see what that looks like. And students will take turns in asking their questions. Now, where this is a little unique is that these are going to be only yes or no questions. You can't ask any question that you want, just so that we can create more standardization of these experiences across the competition and ensure that everyone's getting the same fair experience and the same information. So you're given a total of 11 possible questions that you can ask, and you can ask in total as a group up to 4 questions. And again, these are only going to be yes or no. They will not tell you any other information besides yes or no, but this will provide you with a little bit more information to help you narrow down which case we're talking about. Every participant is given the full list of 40 possible standardized patient illnesses. So you do not have to remember all the names of these illnesses. You will only have to write the number of the illness associated with that illness on your form. So you do not have to have all 40 memorized with you. You'll have that 40 in front of you when you go ahead and take the exam. You're going to select your diagnosis for each standardized patient case and just mark that on the form and turn it in for grading. And again, we will not give you these results until the end. So how this is working essentially is, let's go with the scenario 1. This is going to be student A in your group is going to ask the first question after the scenario is given. And then the second student, student B, can ask their question. The initial student, student A, can ask their final question, and then student B can ask their final question for a total of 4 questions. All answers, again, are going to be just a yes or no. And you'll write
your instrument.
And you will not have to share your answers. This is going to be something that you're not doing as a group, even though there are 2 of you in the room. So this is entirely just an individual score. Remember, we want your participants and all of you to do well, but this is just your score that you're focusing on for this competition. So you're not trying to help out the person next to you. So don't share any answers or say anything out loud during this time, besides the question aloud that you're asking. So then once the second standardized patient you go to, you're going to switch. So now that who is student B will get to ask their question first, and student A will ask their question second, but same scenario. If there is an odd number of attendees at your competition, there is a chance that 3 people could be in your group. But to make sure that things are fair, there's only a maximum of 4 questions still that can be asked by any given group. So how that would work is student A would ask the first question, student B would ask the second, C would ask the third, and then it would go back to student A for the 4th and final question. And then you would still rotate in order for the next SP that you encounter. Now we're going to work through our first one, and this is an image courtesy of AI. I just wanted to make sure I acknowledged that. So I'm going to go ahead and read an SP script for you guys, because again, you won't be able to see any of this written in front of you, but these are the 11 questions that you're able to then ask. So I'm going to go ahead and read this script for you, and you're going to pretend that I am this fine gentleman in front of you on your screen. Okay. Good morning, doctor. I'm only here because my wife insisted. It's been about 10 years since my last checkup, and I felt fine then, so I don't see why anything should be wrong now. She made me bring a list of things she's noticed. For the past year or 2, I've been getting fatigued early in the day, drinking more coffee just to stay alert at work. I'm a masonry operator, by the way. I've also had this constant discomfort in my upper right belly, nothing severe, but it's always there. My wife also says my gut is getting bigger, which is odd since I haven't changed my diet and even quit drinking beer months ago. I still drink a small bottle of whiskey every night, though. She's also freaked me out a few times, saying my eyes look yellow, but they don't seem that bad when I check. Oh, and she wanted me to ask about these little red spider-looking veins on my face, ears, and chest. They're not painful, so I didn't think much of them. Anyway, according to my wife, I'm a tired, yellow-eyed, big-bellied man. I can live with the discomfort, but if there's anything to do to help with the fatigue, that would be great. What do you think is causing all of these problems? So I'm going to go ahead and say that if just anyone wants to put in the chat and put in the chat what question they'd like to ask me, and then I can give you an example of what my response will be. All right, if anyone wants to type in the chat. It's a lot to work through. All right, I'm seeing 11 is, would sending you to our diagnostic imaging department aid in my diagnosis of your illness? My answer is yes. The next question I see is, are your symptoms related to lifestyle choices that I made? My answer to that is also yes. And then the next ones that came in were 6 and 10. So for 6, I'm seeing, are your symptoms related to uncontrolled cell division of a specific cell types? My answer to that is no. And then the final one, number 10, was, would collecting a sample of body fluids aid in my diagnosis of my illness? And my answer to that is yes. Okay. And now normally you'll have the 40 possible scripts in front of you guys, but I'll give you guys a couple minutes to think about it. And if anyone wants to drop in the chat, if they have that list in front of them, if they know which one I'm talking about, go ahead and feel free to put that in the chat, but I'll give you a couple of seconds to work through that. I'm seeing one in there that we've got jaundice in there. That is one of the symptoms that I have, and I see liver cirrhosis. And that is absolutely correct. You did an awesome, awesome job with that. Okay, so good job. So that's how those SPs are going to work. And again, you'll experience 10 of those, and you'll have a full 5 minutes for that full experience. Moving forward with our oral examination, How this is going to work. The rest of the exams are all going to happen on Saturday. This oral exam is going to happen on Sunday. How this will work is that participants are all going to be seated in the frontmost chairs. Your parents, if they are attending, or whoever your chaperones are, they will be invited to attend in the back of the room and observe the competition in real time. How this will work is a moderator will read a question out loud, similar to how I just did with the SP script, and the question will be repeated a second time. After the question is then repeated the second time, you will have 60 seconds to enter a response using an electronic polling system on the device that you brought to the competition. Spelling is totally fine. Don't worry about that. Just make sure that you're putting in an answer. We understand 60 seconds and asking things out loud, things happen there. So don't worry about spelling. How this will work is that you will put in your responses, and once you have gotten 3 strikes, you're essentially out. So after you have entered in 3 incorrect responses, you will no longer gain any more points for the rest of the oral competition. So for example, if you had 3 incorrect responses and there's 10 questions that would be asked then your final subtest score would be that you answered 7 questions correctly. And then that 10th question was one of the third that you answered wrong. So again, 7 out of 10. And at that point, you wouldn't gain any more points for the rest of the oral examination period. These questions are all going to be identical throughout all of the regional competitions. And then again, you will receive this score pretty much right afterwards, because this is when we're going to tabulate your final scores and then provide them to you and announce our winners. So a very exciting final examination for you. Now I'm going to read a question out loud and give you an opportunity to work through this one. I'll repeat it again twice, and then I invite you to put in the chat what your answer is. In the advanced stages of Lou Gehrig's disease, or amyotrophic lateral sclerosis, individuals often experience respiratory insufficiency leading to respiratory failure. This is due to the demyelination of peripheral nerves. Which pair of nerves innervates the diaphragm, and loss of those nerves would contribute to respiratory failure? The question again was, in the advanced stages of Lou Gehrig's disease, or amyotrophic lateral sclerosis, ALS, individuals often experience respiratory insufficiency leading to respiratory failure. This is due to the demyelination of peripheral nerves. Which pair of nerves innervate the diaphragm, and loss of those nerves would contribute to respiratory failure? I'm seeing a bunch of phrenic nerve jumping in the chat. You guys are all rock stars, and that is 100% correct for that question as well. So that is all that I had for you as far as running through the examination. So I'm going to say no questions just to make sure all of our presenters get to go through all of the questions that they have, but I do invite you to put any questions in the chat. So with that, I get the great honor of announcing our first presenter, and stop sharing my screen so that she can go ahead and take over. And this is going to be Dr. Claire Werner, who's going to present our anatomy regional objectives questions interactive review for the day. Dr. Werner got her PhD from Penn State University, where actually Dr. Haggerty and I went as well. So it's a great place to go to learn anatomy if any of you were interested. And Dr. Werner is a assistant professor for anatomy at the University of Pittsburgh School of Dental Medicine. And we're really glad to have her here today. So with that, I'll turn things over to you, Dr. Werner.
Thank you so much, Dr. Pascura. It's nice to see all of you, and congrats to all of you for just being here tonight, being present, and learning and taking advantage of this wonderful opportunity, as well as for those of you who watch the recording later. So thank you all for everything that you've done to put this together, and thank you for coming. So I'm going to get started here. Let me just minimize this so it's not blocking my view. Okay,
Take it away.
do you want to give me a thumbs up if you can see my screen? Okay, awesome. All right, so we'll get started. So we'll kind of first, just a reminder, I know this was previously shown here, but your checklist for your learning objectives can be found at the anatomy website here. So that's kind of where a lot of these questions were based off to kind of fit and match those objectives. We'll get beginning off with our first question here. So what plane of the body is demonstrated in the image below? We've got 5 options here. So coronal plane, sagittal plane, transverse, parasagittal plane, and mid-coronal plane. So go ahead and select your answers. And once we get a good number here submitted, we'll go over it. Looks like we're getting a lot of good answers kind of coming in here pretty quickly. Got a handful still remaining.
All right, I will count down from 5 and you guys will close the poll. Answers will close in 5, 4, 3, 2, 1.
All right, so from what I can see, most of you picked the answer C. All right, and that is awesome to find out here and learn. We can go to our next slide. We can see that C is the correct answer. So wonderful job. Let's kind of talk through that here because we've got a couple alternative planes here that we can address to kind of talk about why we would select transverse in this case. All right, so when we think about a transverse plane, I like to really think about that as showing us a great perspective of how all these structures are located to one another, particularly dividing that body into a superior and inferior part. So if we look at the diagram on the far side of the screen here, we can see that pink division line here dividing into a superior and inferior part of the body. I'm using the word part very specifically. I'll kind of come back to that momentarily. Our next plane, just to identify a coronal plane, we want to consider that would be an anterior and posterior parts, kind of a front and back part or division of the body. You can see that nicely here. Similar structures as our transverse plane. We've got our lungs on either side and our heart in the center here. The third type of plane would be sagittal, and that's going to divide the body into a right and left part. So this can give us some unique perspectives as well of how structures are located to one another. If we look at these images, I'm going to zoom in here just to get a better view. We've got the image here on the left. This is going to be a mid-sagittal plane, so right along midline. But I keep saying part because sometimes it won't be on midline. So this image here on the right side is actually off of midline, but it's still showing us a sagittal cut. So I always like to kind of remind people that we can see that these planes can be conducted on midline or off of midline, so you can get some different views. All right, great job with that question. A lot of good answers coming in there. So we'll move to our next one. Number 2, which organ system is responsible for glands and organs that produce hormones? We've got circulatory, endocrine, nervous, respiratory, or skeletal. So take a moment and send your answers into our poll. Everybody's very quick to answer this one.
All right.
Right.
Since participation is so good, we'll close this one in 5, 4, 3, 2, 1.
All right, wonderful job. It looks like we got almost all of our answers in, and everyone that responded said B, and that is absolutely correct. So, oh man, you guys are off to a great start. So for endocrine system, absolutely, we're thinking about this system kind of really regulating those glands and those organs that are producing hormones for the body. Very widespread, as we can see in this diagram here. Okay, all the way from structures in the brain into reproductive organs, things in the thorax, things in the abdomen. So very, very diverse location. When we look at our other options here that we could have selected, so we've got circulatory system. We want to think about the veins, the arteries, the heart, and how they kind of send blood to structures and then return that blood to the body, or I'm sorry, to the heart so that it can kind of get its nutrients returned. For option C, we've got our nervous system. We want to think about how our nerves are going to supply various structures in the body. So that's a really big topic. We have some other questions on that coming up shortly. Respiratory system, that nice exchange of nutrients, once again, kind of thinking about how its relationship to the heart and how critical that is. And then lastly, we've got our skeletal system, building that framework for the body. So great job, everyone got that one correct. So we'll move to our third question. This one here, we've got the shoulder joint is classified as which of the following? So we've got hinge joint, ball and socket joint, plane joint, fibrous joint, and cartilaginous joint. Go ahead and drop your answers into the poll when you're ready. Such wonderful participation tonight. This is awesome to see.
Great job with answering too. We'll close this one also in 5, 4, 3, 2, 1.
Thank you all. That is phenomenal. So for the most part here, we've got the bulk of our respondents saying option B, and that is absolutely correct. All of you are so prepared. I love to see this. So when we think of the shoulder joint, absolutely, we're thinking ball and socket joint. I've got a couple images here for you when we think about that. If we look at this image on the top left, we can see that head of the humerus. So that's the bone of the arm articulating with the scapula here. All right, and this is a really unique special joint because it's extremely, extremely mobile. It's often compared to being similar or comparable to a golf ball on a tee because it has a very small point of contact here with the scapula bone itself. So another name for this shoulder joint is the glenohumeral joint, so you might hear that as well. And I wanted to kind of just leave you all with one thought about joints, a nice kind of overarching concept. When we think about ball and socket joint, we know it has a lot of mobility, so it can produce almost pretty much every movement at that joint, which is really pretty incredible. And when we think about that, we want to consider the fact that the more mobile a joint is, the less stability a joint has. And those relationships are so important when we think about joints, because you can flip that around saying, okay, well, this joint is very stable, so therefore it's not very mobile. And that's a nice kind of summary to carry with you as you study different joints throughout the body. So absolutely, you all got this ball and socket joint option. So we're going to move here to our next question. All right, question 4. What is the primary function of muscles of the posterior compartment of the arm? So your options being flexion, extension, medial rotation, abduction, or adduction. So once again, go ahead and drop your answers there into the polling option. Lots of answers coming in. Awesome.
All right, we will go ahead and close this poll in 5, 4, 3, 2, 1.
All right. So we've got a little bit more of a mixed response. For the most part, everyone landed on B, which is correct when we think about the muscles of the posterior compartment of the arm. We've got a couple other answers, so let's kind of talk through this here. In general, when we think about these compartments of the upper limb as a whole, so both when we think about the arm or if we're thinking about the forearm, we want to keep in mind that they all have a nice and straightforward function. So if we look at our diagram here, we can see in that darker color, our posterior compartment of the arm. Same here, that darker color being that posterior compartment of the forearm. And in both cases, those are our extensor compartment muscles. So the posterior compartment of the upper limb as a whole is going to generate extension, which is really nice because it's a good way to summarize all of those muscles. We can use that kind of comparing it to the anterior compartment for the upper limb. So once again, at the top of this diagram here, we can see the anterior compartment of the arm, and on the bottom of this diagram, the anterior compartment of the forearm. And once again, in both cases, they have similar functions here. They're kind of lumped together as being flexor compartments. So this is great. We see that nice relationship of a flexor versus extensor compartment to generate these movements of the upper limb. We'll actually talk a little bit later about some muscles that generate some other actions, particularly at the shoulder. So we'll come back to some of these other functions momentarily, but great job. Awesome. So question 5 here, which of the following muscles medially rotates the shoulder joint when it contracts? So we've got latissimus dorsi, trapezius, rhomboid major, supraspinatus, and triceps brachii muscle. Go ahead and enter your answers here into the poll. Got a few responses already. Such awesome participation. This is great.
All right, we will go ahead and close this poll in 5, 4, 3, 2, 1.
All right, so this is great because this is a often commonly challenging thing for individuals to think about as muscle function. So we've got kind of a mix of answers. So we're going to talk through this here. When we look at our option list and trying to think about what can medially rotate the shoulder joint, we want to first try to think about what that action looks like. So medially rotating, kind of rolling that shoulder in an anterior direction. So kind of bringing that shoulder towards the anterior surface of the body. Let me go forward here. The answer that we're looking for in this case is actually going to be our latissimus dorsi muscle. And this can be a really challenging muscle to visualize. It has a very unique pathway compared to a lot of our other options here. So it can be difficult to sometimes think about how it can actually act on the shoulder joint itself. So in general, I always like to think about these muscles, their origin and their insertion. If we think about what side of the body they are on, and if we think about the location of their origin and insertion, we can really think about muscle movement. So let's start with looking at trapezius here because we can see it is fully kind of on that posterior surface from this perspective that we're viewing it. There is a portion we can see from the anterior view as well. But overall, when we look at, oh, I lost my mouse. Hold on, let me go back. Okay. Overall, when we look at its fibers and its pathway, we can kind of see it's originating here from the vertebral column from a medial, it's kind of originating medially and it's inserting laterally. So it's inserting on a lateral point. Additionally, we can also often think about origins of muscles originating more superior and their insertion being more inferior. So those are kind of some general rules of thumb to try to think about muscle movement. It's not always perfect, but it's a good place to start. So when we look at all of these muscles here, we've got our rhomboid major, we've got our supraspinatus muscle. Those, as well as the trapezius, are on the posterior side of the body. So they are not, in this case, going to be medially rotating at the shoulder because of their fiber direction and their origin and insertion. But let's talk about our latissimus dorsi muscle here, because as we mentioned, it's kind of a special, unique muscle. We can see it's originating here on the back on these the spinous process here of the vertebral column, and it's actually going to wrap through the axilla, so through the axilla towards this anterior surface of the humerus. And because of that, if you imagine those muscle fibers shortening, kind of coming closer to one another, it will pull that shoulder joint into medial rotation. Just to give a good comparison, if we look at supraspinatus muscle, that fiber direction or orientation is very linear. So it's not crossing that shoulder joint at an angle that would generate that rotation. So I really, I urge you all to kind of practice visualizing the origin and insertion of those muscles and try to think about if those muscle fibers were to shorten, how would that joint move? It's always one of, I think, the hardest things to try to conceptualize and visualize in your head. All right, our next question here, we've got what structure is indicated by the black arrow? We've got root, gray matter, spinal nerve, and dorsal root ganglion. So go on ahead and select your answer here in the poll. See a lot of people responding very quickly. Got some quick anatomy test takers here.
We have some neuro whizzes in the chat too. We will close this poll in 5, 4, 3, 2, 1.
Awesome. Oh my goodness. Great job, everyone. This is really cool to see because I know neuro is also often a really challenging aspect of anatomy. So when we look at this structure here, a lot of you pretty much were seeing this structure and immediately saying dorsal root ganglion, and you are absolutely correct. So great job with that. I've highlighted and kind of labeled some of these surrounding structures just to kind of talk through their general design and function. So if we look at our structure that we indicated here, that dorsal root ganglion, we can often use that as a great reference point to kind of identify which side of the spinal cord we are looking at. Because when we hear dorsal root ganglion, we're thinking it's on the dorsal side of the body or dorsal side of the cord. So if we take a look, we'll follow that dorsal root ganglion. It's leading us to that dorsal root. We can kind of see those rootlets here that are leading us to that dorsal horn. All right, and that's kind of all highlighted in red because those will contain sensory related information, whether it's fibers or cell bodies. If we look on the opposite side here, we've got our ventral horn leading us out to our ventral root here. And now we can see those are all in black. So those are our motor components. Once again, motor, cell bodies, or fibers. And now you can see I've kind of tried to combine those colors. We've got the spinal nerve forming, and that will give us a couple other named components. And the spinal nerve will carry both fiber types. So that's always a really key feature to think about when we think about spinal nerves carrying motor and sensory information to or from the necessary regions of the body. All right, great job. Lots of awesome neuro anatomy coming into play here. Y'all did wonderful. Our next question is, which region of the brain is primarily responsible for coordinating movement? So we're back to neuro again here. We've got cerebellum, frontal lobe, occipital lobe, parietal lobe, and temporal lobe. Go ahead and drop your answers in the poll.
Participation's good again. You guys are doing a really great job so far. We'll close this one in 5, 4, 3, 2, 1.
All right. Great job. So we've got a couple different answers here. For the most part, we were seeing people respond A. So let's talk through this. A is absolutely correct. So way to go, everyone, with their neuro knowledge tonight. So when we think about these different regions of the brain, once again, very complex structures, we want to think about blocking my view here. Let me move this to the top. When we think about these structures of the brain or regions of the brain, generically speaking, in this kind of image on the left, we can see our frontal lobe, our parietal lobe in blue. We've got the temporal lobe in orange and that occipital lobe. In this image, it's not including the cerebellum, but we can see that in this other image here in purple. All right, and when we think about the cerebellum, we always want to keep in mind it's related to movement, kind of coordinating movement, as well as preventing motor error. So it's got kind of this nice learning component to it as well for producing movement. When we look at these other regions, when we think about the frontal lobe, it's going to be a lot of that planning, decision-making type of function. Think about occipital lobe on the very back side or posterior side of the brain. We're thinking about vision. So that's another nice kind of landmark there. It's going to be just superior to that cerebellum. Our last two lobes here in blue, we've got that parietal lobe. When you're thinking about parietal, it's going to be kind of a combination of movement and touch, okay, kind of being the bulk of what you'll see there. We'll also see temporal lobe, a lot of processing of memory and emotion. So very complex regions here of the brain. Y'all did a wonderful job. So we will move here to our next question. Oops. Okay. All right, number 8, we've got deoxygenated blood enters which chamber of the heart? So left atrium, left ventricle, right atrium, and right ventricle. Go ahead and drop your answers there into our polling system. The heart being a really nice, important structure to be studying. So this is great to see all of you answering.
And we'll go ahead and close this poll in 5, 4, 3, 2, 1.
All right, looks like everyone put an answer in. Thank you. That is awesome to see. Love that. So for the most part, we have everyone answering for C, and that is absolutely correct. This is a really great thing to kind of think through when we think about how blood is passing through these different chambers of the heart, and therefore then how it gets to the body once it's now kind of regained all those nutrients. So absolutely, when we think about blood entering the heart, it's going to be deoxygenated, right? So nutrient poor, oxygen poor blood entering that right atrium, so the right side of the heart. It's going to then pump through that right atrium, kind of through the tricuspid valve into the right ventricle. I've also kind of left that in blue because that will also be prior to it reaching the lungs. So it's still nutrient poor, oxygen poor blood. From the right ventricle, kind of pumping through the pulmonary valve, it will then attribute to the lungs. So that's kind of where we can see in this diagram now we've brought that blood into the lungs, and now it can get kind of resupplied, right? Get that oxygen back into the blood, nutrients back into the blood, and then enter the left atrium. So you'll see then when we think about the left side of the heart, we like to kind of usually depict it in red because that's going to be the side of the heart that has that oxygen-rich blood. All right, so left atrium will be receiving that blood back from the lungs. It will then pass through the bicuspid valve to that left ventricle, and the left ventricle has the magical job of pushing it through the aortic valve to go into the aorta and attribute blood to the entire body. So that's kind of in general, you want to think about the right side of the heart being deoxygenated blood and the left side of the heart being oxygenated blood. All right, great job, everyone. So moving along, kind of sticking with that general theme here of the heart, what artery is indicated by the black arrow? We've got right coronary artery, left coronary artery, anterior interventricular artery, and posterior interventricular artery. I would say the heart is probably the hardest organ to orient yourself to, so take your time. It's always a challenge to figure out what side or surface you're looking at. Got a lot of awesome answers coming in here.
We will close this poll in 5, 4, 3, 2, 1.
All right, great. So as I mentioned, this is definitely the hardest organ to orient. Looks like a lot of you did a really great job with this. We've got a couple different answers, so we'll talk through this. When we look at the heart, it's always tricky to tell. Are we looking at the anterior surface? Are we looking at the posterior surface? What aspect of the heart are we looking at? So a couple landmarks that I like to use, actually, let me go back to the unlabeled image here. If we are looking at this aspect, can you see my mouse? I just realized it might have disappeared. Okay, I'll actually grab my laser pointer for this here. So when we look at this surface of the heart, we can see that right atrium here very nicely, and it has this nice little projection coming off of it that can be a really good landmark for you to look for and kind of know, okay, we're looking at this anterior surface. And this is the right auricle. Okay, so they call it auricle because it's ear-shaped. Sometimes they're more clear than others, but we can see that nicely. We can also see all of these vessels here that we've kind of considered as being these large, great vessels of the heart, bringing blood to or kind of towards the lungs, or bringing it back from the lungs into the heart itself. So the image here that identified the artery here that we were identifying on the last slide was the left coronary artery, which a lot of you had selected, so awesome. When we think about these coronary arteries, we know that they supply a couple of different critical components of the heart, so it's always important to think about what branches they give off, because it can be really related and tied directly to clinical correlates. So if we see that right coronary artery here on that anterior surface, we can see that's going to be one of our main coronary arteries. Our left coronary artery here is very short before it immediately splits, giving off that circumflex branch and that anterior interventricular branch. Some of you clinically will hear it being that left anterior descending branch. So put both of those options there for you. It's really nice when you hear anterior interventricular, you know, it's on the anterior surface and it's going to be dividing those ventricles. So it can be a great landmark for you. As well as this right coronary artery, it's a nice landmark to divide both of those right chambers of the heart. So some really great features here when you're trying to identify what surface you're on. I also just wanted to highlight from a clinical perspective, something really interesting to think about is that some hearts will be right coronary artery dominant and some hearts will be left coronary artery dominant. So different times of blockage of these vessels can mean different things for different people. And that's a really interesting human variation feature to take into consideration in the clinic. All right, great job. So we'll move to question 10. This is great. We had a little preview to this earlier, so I'm excited to see what all of you say. We've got what primary nerve is innervating the diaphragm. We've got Purkinje fibers, intercostal nerve, vagus nerve, or phrenic nerve. So go ahead and drop your answer here in our poll. Lots of fast responses. Such awesome participation tonight. Thank you all so much.
We'll do this one a lot faster since you guys showed us you knew what we were talking about before, but we'll close this one in 5, 4, 3, 2, 1.
All right, great job, everyone. This is awesome. It kind of brings us back to a previous point from Dr. Pesquera. So it's always good to hear things in different ways, see some images so we can tie this all together. You all did a really good job with this question. We're thinking about the diaphragm. We're absolutely thinking about phrenic nerve being that primary nerve to innervate this muscle. I always have heard and utilized the phrase C3, 4, 5 keeps the diaphragm alive. C3, C4, and C5 are the components comprising this nerve, and you can see it's going to descend through the neck, through the thoracic cavity, and all the way down to the level of the diaphragm. So this nerve has quite a path that it takes, and a really great structure to see when you're in lab. So hopefully you all get a chance to see that as well. All right, great job. So question 11, oh, sorry.
So I think we could do this one last question, and then after this one, I just want to give you guys a heads up. We'll shift gears to some histology and embryology, but doing great so far.
Thank you. Perfect. All right. So our last question, we've got what named region of the lung receives the arteries, veins, and bronchus? Got apex, base, root, hilum, and lingula. Lots of fast responses coming in here. Great to see.
We'll close this poll in 5, 4, 3, 2, 1.
All right. Great job. We've got kind of a mix as well. This is always a little tricky because there's a lot of features here that you'll see on the lung. So for this one, froze on me. Sorry. There we go. For this one here, we're going to be looking for the hilum. So when we think about the hilum, this will be that region on the lung that will receive all of these different structures, the arteries, the veins, and the bronchus. Just a summary slide for all of you on this. When we think about that hilum, right, just as I said, that site or surface on the lung receiving these structures, this will be on that mediastinal surface. When we hear mediastinum, we want to think about the heart. So it's that surface that's facing the heart. And then when we think about the root, just to kind of get a comparison, the root is those group or collection of those structures that are entering this hilum or surface of the lung. All right, so we've got those pulmonary veins, pulmonary arteries, and the bronchi. In general, you'll see those structures kind of towards the center of this surface of the lung. And when we think about those pulmonary veins, we'll see they're usually more anteriorly located and inferiorly located. We'll see those pulmonary arteries being more superiorly placed, and then the bronchus more posteriorly placed. And if we think about those structures and kind of where they're located, think about the trachea and how much deeper that is, we can start to visualize how they reach the lung. So thank you all so much. I really appreciate it. I will stop sharing here so that the next presenter can share their screen. Great job, everyone.
Thank you so much, Dr. Werner. I think you did such a great job in breaking those things down too, and making that really nice and clear and digestible. So really appreciate your time for you coming on tonight and taking the time to chat with us. All right, next up, you have seen him before and you know him, you love him. We've got Dr. Alexander Macnow from Thomas Jefferson University. And again, Dr. Macnow works with Dr. Haggerty. And again, just did such a great job, I think, last time that we had all met together, and really excited to go over our review questions with him again tonight.
All right. Hello, everyone. Nice to see you all, at least virtually. I'm going to be going over some of the histology questions here. Now, I'll note the questions that I have here really vary in terms of difficulty and style. So at the very beginning, Dr. Piscura talked a little bit about first order and second order questions. Some of these will be sort of straight ID. Other of them will be second order. Some will even get a little bit into the clinical application world. But all of them should still map with the learning objectives you have. You'll see that I'll highlight those learning objectives as appropriate with the questions, and should still be able to be solvable or figure outable with first principles that you would be expected to know for anatomy. So again, all the learning objectives are listed online. This will be on YouTube, so you can always copy over the link from there or go directly through the Anatomy website. And we'll dive right into our first question. So we have here, which of the following is seen exclusively in the muscle tissue shown? We've got an associated image. So take a moment to work through that, and then we'll chat about it. We've got a nice mix of answer choices here, which is a good thing to see. It gives us an opportunity to provide some points of clarity. Okay. So it does look like everyone has responded here, at least based on the count that I'm seeing here in the poll. Correct me if I'm wrong.
I'll go ahead and close it now.
Okay, sounds good.
Okay.
Yes, we've got a diversity of answer choices, so that is great to see. This is a second order question. So our first task, of course, is going to be figuring out, well, what is the muscle tissue that's being shown? We've got three options predominantly there. So we'll talk through that. And then we got to figure out, well, which of these actually are specifically associated with this particular type of muscle tissue. So that's a second order question. We've got two steps to really get to the answer choice. So let's start with that first part and determining, well, what is the muscle tissue? And one of the things I'll talk about at the end strategically is being able to identify or to think about what are the things that help us to compare and contrast things in the same category. I talk about this constantly with my medical students because as a pathologist by training, much of my job is figuring out, all right, if someone has cancer, how do I figure out of the hundreds of types of lung cancer, maybe 5 main types, but hundreds of different kinds of lung cancer, How do I know which one it is? What features tell me it's one versus another versus another? Or if someone's going to start someone on a medication, they have to figure out, well, how do I compare and contrast what type of medication makes the most sense for this person's diagnosis? And so being able to figure out how do I compare and contrast things in the same category, what do I look for to make those comparisons is really important. So I'll have a summary table at the end described from one of the other review sessions, but if I'm looking at muscle tissue, things I'm looking for are, well, are there striations? Do I see an indication that there are stripes of alternating color or nice parallel lines to indicate the presence of sarcomeres? That's going to tell me some good information. I'm going to be looking at nuclei. Do I see many nuclei all stacked together? Are the nuclei toward the center of the cell, toward the periphery of the cell? I'm going to be looking for, is there any branching, or what are the shapes of the cells that I'm looking at? And those might help me. So here, let's walk through some of those principles in this question. Oh, there we go. So here, ultimately, we're going to be looking at cardiac muscle, but how do I know? Well, I'll look for things like the presence of striations. So again, here I can see all these nice parallel lines, which will be my contractile apparatus. And I can kind of see cutting across that these alternating bands of light and dark. Now, if you're a little less than convinced, that's okay. There are other features that also still help us here. So the nuclei, when I look at the placement of these nuclei, they tend to be hanging out pretty close to the middle of a given cell. Well, that's going to help me know cardiac muscle versus, say, skeletal muscle, where in skeletal muscle, my nuclei are peripheral. They're toward the edge of the cells. And I may also look for things like, again, branching. And here, there's not a huge amount of it, but close to where I've labeled this intercalated disc, I see some branching right there. There's a Y shape of branching across those cells. Or if I go up here, again, I can see this branching, splitting essentially of cells into separate branches. So those are all things that would tell me I'm looking at cardiac muscle. And a unique feature of cardiac muscle would be intercalated discs. And we'll talk about their function as well. But that would be our answer here. Now, for the learning objective, as I mentioned, I'll include for each question what the related learning objective is. I'm not going to read it verbatim. You don't need me to waste your time reading that, but just to sort of prove and to demonstrate how this remains in scope of the anatomy. So this then links to one of the sessions by Dr. Oriana, where we looked at the musculoskeletal system, and again, comparing and contrasting these three types of muscle. So as I mentioned before, on the one hand, you can just sit there and memorize everything that's in this table. But I think what's important is also recognizing, how do I use this information? So in something like this, where I have to do some histological identification, I have to identify what I'm actually looking at before I even take that next step to answer something about the content, something specific to that tissue. I want to know the things, again, that I'm looking for. And those are mostly highlighted in this table. Again, things like, are there a single or multiple nuclei? Where are those nuclei positioned? What do the cells look like in terms of their shape? Whether they're just sort of straight cylindrical tubes like skeletal muscle, the branching pattern that we see in cardiac muscle, or what we call fusiform, sort of twisted or elongated in shape, kind of tapered at both ends. Do I see an appearance of striation? That's true for both skeletal and cardiac muscle, but not for smooth muscle. Where in smooth muscle, instead, I would have organization into contractile fibers and dense bodies, but not organized in these nice alternating bands. As well as, again, what types of innervation exist to those different types of tissues, whether that's somatic or voluntary contraction in skeletal muscle, or autonomic and therefore involuntary contraction in cardiac and smooth muscle. So again, I haven't necessarily gone through every single entry that's in this table, but the point being, when I'm looking or thinking about muscle, these are the types of things that I would want to compare and contrast. Now, if I jump back to the previous question, I would also point in here as someone who taught test prep for a long time. I worked for Kaplan Test Prep as one of the directors of MCAT programs, which is the exam that pre-med students take when they're applying to medical school. So I spent a long time in that world, and so I've done a lot with practice questions. I would also give you as a word of encouragement when you are practicing with practice questions to, sure, determine what's the right answer and why is that the right answer, but also make sure to quiz yourself on the various incorrect answer choices and where those would apply, where the correct answer, or where that would be the correct answer. So reminding ourselves maybe what dense bodies are, which are, at least in smooth muscle, where that contractile apparatus, the fibers, are attaching to. They take the place of Z lines that exist in striated muscle. Myotendinous junctions. Well, myo is muscle, tendon is tendon. So this is going to exist only for skeletal muscle, where muscle fuses into tendinous tissue to connect over to bone. Neuromuscular junctions, of course, will exist in all muscle types. So of note, that is a feature of cardiac muscle. Sure, we would have autonomic fibers firing onto cardiac muscle, but that's not exclusively seen there. That is a feature, again, of all three types. And finally, sarcomeres, we do see that in cardiac muscle, yes, but we also do see that in skeletal muscle. So maybe a little bit of a tricky one to start with here, where we're trying to categorize and identify these details, but being careful with that wording. Here we were looking for something that exists in cardiac muscle and not in smooth muscle and not in skeletal muscle, and that would be intercalated discs. Just to mention, by the way, the content there, and then we'll move to our next question. Intercalated discs are really important in cardiac muscle because they basically connect the cardiac muscle cells to each other, both physically and electronically. And so they will have what we call desmosomes that basically mechanically link the cells to each other, so they act physically as a unit, as well as gap junctions, which are small tubes that allow ions to go from one cell to the next, and therefore allow the electrical signals that are traveling through that cardiac muscle to seamlessly go from cell to cell to cell, allowing for uniform contraction. All right. So we're going to get even maybe slightly more complicated, but you can do it. I've got faith. Take a minute, we'll read through this question, and we'll answer it. So the disease MS, or multiple sclerosis, is due to autoimmune attack on oligodendrocytes, leading to death of these glial cells and loss of their function. Which of the following would be an expected finding in multiple sclerosis? And then we got to decode what's going on in these answers. So let's take a minute to think through this, and then we'll chat about it together.
All right, this poll will close in 5, 4, 3, 2, 1.
All right, so clearly a very clinical question here. This is something that when we're thinking about MS and the pathogenesis, the disease and how it's formed in MS, and really the symptoms that someone experiences there, we have to know a little bit of how the disease works, what we would call its mechanism. So here again, we basically have the oligodendrocytes being killed off here by attack by the immune system. And so the question behind the question here, of course, is, well, what do oligodendrocytes do? What is the function of those glial cells? And we may remember that oligodendrocytes are one of the two cell types that we'll see in the nervous system that develop myelin, which is going to be important for us as insulation around the cells. And we'll talk about its specific role physiologically to tie it into this question too. But there's another cell type as well that does that function, which would be Schwann cells. So we'll have to differentiate, well, what's the difference between an oligodendrocyte and a Schwann cell? They're both forming myelin. Myelin is this fatty insulation on the outside of the axons of the neurons that are in that central nervous system. And we may remind ourselves that there are little spaces between where those areas of myelination are called nodes of Ranvier. The importance of that system, the myelin, the nodes of Ranvier that are scattered down that axon, are that they permit what we call saltatory conduction, which is saltar, if anyone speaks Spanish, is the verb for to jump. And the idea here is basically that the signal is functionally jumping from node to node to node, and it's able to go down that axon much, much, much faster than if that axon was fully unmyelinated, and basically every single tiny segment of that axon needed to individually fire that action potential. So, okay, that means it's going to have something to do with the speed of conduction. I'm hopefully looking for that in the answer choice. And then I'm also thinking to myself, well, am I thinking about Schwann cells or oligodendrocytes, and what again are the differences between them because they both do myelin? And the answer to that is location. Whereas oligodendrocytes are specifically seen in the central nervous system, Schwann cells are specifically seen in the peripheral nervous system. So here again, oligodendrocytes, their function is myelination. In terms of appearance, they often are described as having a fried egg appearance. And I see that. There's sort of this central nucleus that's like the yolk, and then this nice myelin-containing fatty material essentially around it. So that's kind of like the whites of the egg around it. When people have tumors of these cells, we often describe it as having a, again, fried egg appearance of those tumor cells. And again, in terms of their function, they're myelinating these neurons, allowing for those very small areas of bare axonal membrane where the signal basically jumps from that node to node to node. So if those oligodendrocytes were killed off, we would expect that process of saltatory conduction to not occur, and therefore for conduction velocity to be slowed down. So our answer here is going to be D. We would have slowed conduction velocity here in the central nervous system specifically. There are other diseases related to any and all of these other findings too, but those would be affecting different glial cells. So let's make sure that we identify those glial cells. Here's our two learning objectives that are both kind of related to this question when brought together. And this comes from the session that I had done about nervous and supporting systems and the major glial cells that we're seeing there. So in the CNS, again, we have those oligodendrocytes already described. We'd also have astrocytes that are important for physically supporting the neurons. They also, again, establish that blood-brain barrier, being very selective about what can actually get in to the neurological tissue and what can't. Here I had pointed out during our session that this is actually a small blood vessel that they're lining the two sides of. And you can kind of see there's like a tube cutting through there. That's those axons, or sorry, those astrocytes sending little processes down to that blood vessel to control what's able to get in and out of it. We see some microglia. Those are the phagocytic cells, the ones that'll come and gobble up dead material or debris of the central nervous system, and ependymal cells that produce cerebrospinal fluid. Over on the other side, we've got some cells of the peripheral nervous system, Schwann cells serving the function of myelination there, and some satellite cells, which we would see specifically in ganglia. Ganglia are collections of neuronal cell bodies outside the central nervous system, so in the peripheral nervous system. And satellite cells help to electrically and chemically basically balance those neuronal cell bodies and keep them nourished. Okay. Let's go for maybe more of a straight ID kind of question here. So here's question 3. Which of the following is the most accurate classification of this structure seen in the ovary?
Alrighty, we will go ahead and close this poll in 5, 4, 3, 2, 1.
All right. So again, we've got to compare and contrast different follicular structures that exist within the ovary here. Some of them are follicles proper, other things that are sort of left behind after ovulation, remnants of follicles. And how do we tell them apart? Well, things again that I would look for here are how many layers of cells are around the ovum. So here, just to identify, the ovum itself is this giant honkin' cell right in the middle. You see its nucleus here, somewhat pale. You see plenty of cytoplasm here because, again, once fertilized, that's going to be where all of the organelles functionally are going to be for that developing zygote. So lots and lots of cytoplasm there. That whole thing is just the ovum. We have a layer around it, which we'll label in just a moment, called the zona pellucida. And then around that, we've got a variable number of granulosa cells. The number of layers of granulosa cells is important for us. That'll help us identify, say, the differences between a primordial follicle and a primary follicle. I'd also be looking for the presence of an antrum. The antrum is the large fluid-filled space that we would see that emerges from within those granulosa cells and basically becomes kind of the predominant feature when we look at late tertiary, or what can also be called Graafian follicles, right before ovulation. So here I look at this and I see a couple layers of granulosa cells, more than just one. I don't really see a notable antrum. I'm seeing the very beginnings, maybe, of these granulosa cells just starting to come apart from each other to maybe get ready to start forming that antrum. But I couldn't call this something that has a full antrum here. And we'll see an image later from, again, the review session to help get that comparison. The other structures mentioned in these answer choices, a corpus luteum or corpus albicans, are what are left behind after ovulation has occurred, specifically from those granulosa cells and other support cells surrounding that called theca cells that basically help to produce a lot of the hormones that get the endometrium ready for implantation. Or once that corpus luteum breaks down, it becomes what's called a corpus albicans. Notably, by the way, just to bring in some of the language here, that changes from having a yellowy color to a whitish color. Corpus luteum means yellow body, and it's colored yellow because it's producing fat-based hormones. Whereas corpus albicans, alba means white, that means white body, and basically it's just a little tiny nodule of scar tissue, which scars generally have kind of a whitish color to them as well. So, okay, that's a lot of blabbing about the content, but what's the actual takeaway? I've got a few layers of granulosa cells here. I don't really have a notable antrum. That is going to be placing me in the world of a primary follicle. If I had a little bit more established antrum, I could maybe say a secondary follicle, but I definitely am not yet late enough in the process here to have a single dominating fluid-filled antrum, which would be the major feature for a tertiary follicle. So that's that ovum again mentioned before, the zona pellucida. We've got a couple of layers of granulosa cells surrounding that. We don't really have a notable theca layer yet. We don't really notably have an antrum yet. That places us here into the world of a primary follicle. So there's our associated LO. And this again comes from the urinary and reproductive system review. Just to recapitulate some of the notable follicle types, primordial follicles are the earliest form. At the time of birth, an individual is born with all of their ova at the level of being a primordial follicle. And they are basically the ovum lined with an incredibly thin layer, squamous layer of granulosa cells, which at that point we really just call follicular cells. We go to an early primary follicle when those granulosa cells start to plump up and start to get multiple layers to them. We move into a secondary follicle when an antrum, that fluid-filled space, begins to form. And finally, to a tertiary follicle when that antrum is really that predominating feature that we're seeing histologically. Again, around that, there are also connective tissue layers that are a little harder to detect from the background, but these spiraling theca layers around the secondary and into tertiary follicles would be hormonally responsive tissues, and that also are helping to produce hormones that support the reproductive tract. Finally, that corpus luteum and corpus albicans mentioned, this is after ovulation. Basically, the granulosa and theca cells turn into hormonally producing cells to help basically with the back half of the menstrual cycle and preparing the endometrium for potential implantation. If that doesn't happen, that corpus luteum will scar down and become a corpus albicans, or a white body, that little nodule of scar tissue left behind. Okay. Let's take a look at another clinical kind of question. So patients with a mutation in the BTK gene have a severe immunodeficiency. This is because BTK is responsible for the maturation of agranulocytes with small dark nuclei and scant cytoplasm that mature in the bone marrow. Well, what cell type is that? What is the function of that cell type? First, you got to figure out what type of cell we're talking about with all that blathering on.
Okay, this poll will close in 5, 4, 3, 2, 1.
Okay. So we've got basically a vocabulary question as much as anything here. We got to decode what the heck that middle sentence is saying. So agranulocytes. We have three major types of leukocytes or white blood cells that are called granulocytes. We've got two major categories that are agranulocytes. Granulocytes have granules in their cytoplasm. Those are neutrophils, eosinophils, and basophils. Agranulocytes well, don't have granules in their cytoplasm. And so those are monocytes in circulation and B cells and T cells, which are our two major types of lymphocytes. Here, if it's going to be in that category of monocytes or lymphocytes, we then look at the remainder here. Small, dark nucleus, scant, which means little bit of cytoplasm. We're going to be talking about a lymphocyte then. Particularly that cytoplasm gives that away. Monocytes have a lot of cytoplasm to them. Lymphocytes, in contrast, are basically just a nucleus with a thin little rim of cytoplasm around it. Then we actually get even a little more information, specifically that they mature in the bone marrow. And that'll help us distinguish between B cells and T cells, the two major types of lymphocytes. Specifically, B cells mature in the bone marrow. People often say B for bone. If we're being really technical and pedantic, it's actually for the bursa fabricius because it was originally discovered in birds, which have that organ. But for all intents and purposes, B can be for bone because they mature in the bone, and T is for thymus, which is where the T cells would be maturing. Then we have even another step. Well, what do B cells do? And B cells, in terms of their function, hopefully we remember, their major function is going to be to first have on their surface and then be able to secrete immunoglobulins or antibodies, which will be helpful for us to respond to certain kinds of pathogens or infectious organisms. B cells specifically will turn into plasma cells when they basically become antibody-producing factories. And so that would match us here with answer choice C. There's our learning objective. And to reassociate again with the review where we saw this, basically we went through the different major types. Well, I did not, but Dr. Frank had gone through the major different cell types here that we had and the major classifications that we had therein. Here again, we're looking at B lymphocytes in particular. Right, I'm hoping maybe I have time for one more question here. Is that good? Great, I'm getting a 5-minute warning, so I think we're good. So here we're being asked, which of the following mechanisms of secretion is being shown in this tissue? Let me give you a small strategic point. See first if you can just figure out, well, what kind of secretion is this? Sort of the first half of the question, and then see if you can figure out the back half. Or you could start at the back end too, but you've got sort of 2 parts in each answer choice, so maybe that helps.
Okay, we will close this poll in 5, 4, 3, 2, 1.
Okay. So first thing, we got to figure out our type of secretion. Let's make sure we know the wording here. Apocrine secretion being where essentially the secretory product comes off in these pinched vesicles, or these blebs basically that come off the parent cell. Holocrine secretion is rather rare, but holocrine secretion is where basically the whole cell just falls apart and becomes the secretory product in that process. Miracrine secretion is sort of the more standard type that we're usually thinking about, where a vesicle would fuse with the membrane and spit out the contents of the vesicle, but it's not that the entire vesicle goes into the lumen or the duct. It's basically just that the contents inside spill into that space. So here we got to take a look at the image. Here we've got relatively pinkish colored cells. We would call those eosinophilic cells, which is often seen with this particular type of secretion as well. But I think most notably, there's some areas when I look in the lumen or the hollow center, the duct area of these glands, where I see these little spheres of basically what kind of look like the cytoplasm next to them. So those are going to be basically these secreted vesicles. That's telling me this is going to be apocrine secretion. Beyond that, I have to figure out, well, where does that occur? And so here, fortunately, I did not give you 2 organs that both undergo apocrine secretion. You didn't technically need to identify the specific gland type, but we would note here that apocrine secretion is one of the standard forms seen from at least one of the 2 types of sweat glands, whereas the islets of Langerhans are importantly the endocrine parts of the pancreas. And in contrast to exocrine glands, which secrete things into a duct, and apocrine, holocrine, and miracrine secretion would be classic examples for exocrine organs, endocrine organs don't have that same pattern of secretion. They secrete directly into the bloodstream. And we could maybe say miracrine is sort of our closest there, where we would release the contents of those cells into the bloodstream, but we wouldn't have these whole basically blebs or chunks of cells coming off. So here, just to highlight that, there are those secretions again from these pinched off little portions of cell. This is going to be again specifically from a sweat gland. And this again came from the fundamentals of anatomy, histo, and embryology here, where we had a comparison and contrast essentially of those three types of secretion. Okay. Okay, I'll throw in one last real quick question here if I've got 2 minutes. Awesome. So I got one more for you. In the process of chondrogenesis, which of the following cell types differentiates to form the chondroblasts from which hyaline cartilage is made? So make sure we know what step we're at in the process here. We're asking who gives rise to the chondroblasts.
You guys are really speedy with the participation on this one, so we'll wrap this one up a little bit quicker. Polls will close in 5, 4, 3, 2, 1.
Awesome. So here, yeah, we're seeing some good answers. And basically, we have to remember in chondrogenesis, we are forming the chondroblasts, which are the cells that actually initially create the cartilage, from what are called mesenchymal cells. Mesenchymal cells are basically really early connective tissue precursor cells. They go and form the vast majority of connective tissue type cells in different parts of the body. Specifically in areas that will become cartilage, they will first turn into chondroblasts. And from there, chondroblasts will mature and they will multiply and they'll produce the extracellular matrix that is cartilage, or at least the extracellular part of cartilage. And they'll ultimately mature into chondrocytes, that nomenclature existing in a lot of places in the body. Blasts are often the immature or young version of a cell line. Cytes are often the more mature or later version in that cell line. So here, chondroblasts will become chondrocytes, but the chondroblasts themselves came from mesenchymal cells in the first place. And that again came from that musculoskeletal system session, where here we're looking at the very first step from mesenchymal cells to chondroblasts, which again then produce that matrix and become the more mature chondrocytes by the end. That's going to be it for me. Thank you very, very much for your time. If you felt these questions were difficult, do not feel deflated. Again, my goal here was not necessarily to be asking questions that are purely just at that Anato-Bee level, but to show some of the application, hopefully, and maybe identify for you a couple places to say, oop, maybe I want to go back and learn a little bit more about this before I go in for the competition. So good luck. I'm sure they'll all do fantastically. If there are any other things that we can help with, don't hesitate to reach out and ask.
Thank you so much. Again, just really appreciate all your time and very thorough explanations with everything that you go through. Just to touch on the difficulty that he had gone through, I think for the written exam, some of those questions might have been a little bit harder, but for the oral exam, I thought that a lot of them were very appropriate. So this is a great way to give them sort of that spectrum of kind of the range of difficulty you can see depending on which examination you're taking in the final competition in the oral round there. So thank you so much for giving them an extra taste of what that is, and again for encouraging them to keep moving forward with their studies. So
My pleasure. Thanks.
thank you so much. With our final presenter, I'm happy to introduce Dr. Troy Camarata from Baptist University College of Osteopathic Medicine. And I think I read online, Dr. Camarata, you are one of the founding faculty actually at this College of Osteopathic Medicine, which is really awesome. And just in looking up more stuff that you were doing, you did some work with some zebrafish. And I think that some of our students, if you could just briefly touch on that really quick, I think our learners would find that really cool.
Absolutely. So yes, I'm a founding member of, this is actually my second founding of a medical school. So we just set our inaugural class in August. So we're rolling through our first year and all the wonderful challenges that come along with that. My research background is in developmental biology. So I've worked with zebrafish and mice and chickens and turtle embryos and all these wonderful creatures to understand cell differentiation and other aspects of development and organ development, so kidney and heart and limb and all these awesome things that that form during embryogenesis.
That's awesome. So nobody better than to talk through embryology than Dr. Camarata. So thank you again for joining us, and the floor is yours.
Excellent. And then if there's questions, just pop them in the chat, and I'll rely on my other colleagues on the call to kind of address those as we go through. So let's get this to work. Okay, so our first question. So an individual who's unaware that she is pregnant has continued to smoke a pack of cigarettes a day for the last 2 months. Her last known menstrual period was 5 weeks ago. So which of the embryo's organ systems might most be affected during this time?
All right, we will close this poll in 5, 4, 3, 2, 1.
So the key here is our 5-week time period. And this is the difference between sort of embryonic age and gestational age, right? So we're looking at gestational age because that's the information we've been given as far as that when that last menstrual period began. So we're at this 5-week period, and this is just simply connecting that time frame to what organ system is developing. At the 5-week period, we're more concerned with our GI system. So hopefully that makes sense for everyone. And then you can see obviously the different development timeframes for our other systems that are listed there. Okay, so we will move on to the next question. So approximately 5 to 6 days after fertilization has occurred in the uterine tube. The embryo arrives in the uterine cavity where fluid is absorbed, separating the cluster of cells into 2 distinct and specialized groups of cells, the inner and outer cell masses. What specialized cells are derived from the outer cell mass?
This poll will close in 5, 4, 3, 2, 1.
All right, so our correct answer would be B. So our outer cell mass is going to be these syncytiotrophoblasts and cytotrophoblast cells. So they're labeled here in this image. Our inner cell mass is going to give rise to our embryo proper. And then our outer cell mass are these extra embryonic cells. They're involved in implantation, especially those syncytiotrophoblasts, and creating the placenta-derived tissue. So again, just kind of differentiating between your inner and outer cell masses and what they will eventually become. All right, so let's move on to our next. So an important molecular signal, signaling structure of the mesoderm is the notochord. So sonic hedgehog is produced by the notochord during week 4 post-fertilization, and it induces the formation of which of these structures and tissues?
You guys are doing great. We'll go ahead and close this poll in 5, 4, 3, 2, 1.
All right, so looks like most people chose B, and that is the correct one. So the key here is just remembering your orientation of where things are in three-dimensional space. So our notochord is going to be just below that neural plate, and it's going to express on a sonic hedgehog, which is then going to go back and communicate to that neural plate and induce these morphogenic changes to create that neural tube. So you get folding of that neural plate, you get cell shape change to create that neural tube. And so that would be the correct response here. All right, so our next question. So failure of the caudal neural pore to close completely can result in a failure of the vertebral arch to completely close at the end of week 4 post-fertilization, when both the meninges and spinal nerves are found outside of the vertebral spine. The condition is known as what?
Okay, this poll will close in 5, 4, 3, 2, 1.
Okay, so we have some variability in our answer choices, and that's okay. So the correct answer here is myelomeningocele. So really the key is, so for answer C, this is when, so the entire spinal column is open, so that would be C. So that's not what we're talking about here in the question. In the question, the key was both meninges and spinal nerves. And on this image, you can see that is classic myelomeningocele. And that's how you differentiate it from just meningocele, is the fact that we have spinal nerve involvement. Spina bifida occulta is very mild. It is not going to really involve the meninges and certainly not the spinal nerves, as you can see within this image. So again, just kind of review some of that material and kind of what are the key features of each one of these types of neural tube defects. And again, anencephaly, no one chose that one, but that's again where a large portion of the skull does not close properly. But that does not relate to our question here. So again, just go back and review some of those to make sure you understand what those key characteristics of each neural tube defect is. All right, so an embryonic derivative of the somatic lateral plate mesoderm forms the skeletal muscle of the trunk and diaphragm. The major lateral plate mesoderm contribution to the diaphragm is which?
Alrighty, this poll will close in 5, 4, 3, 2, 1.
Okay, so it looks like the vast majority chose D, and certainly if one of my colleagues wants to have a better explanation for this than I would, I'm all for it. But yes, D is that correct answer. And so just kind of reviewing this image quickly, you can see the septum transversum, so that's going to give rise to our diaphragm, while we have our other options, our pericardioperitoneal or our pleuroperitoneal membranes. So these are going to help separate some of our cavities between our thoracic cavity and our abdominal cavity and our structures within there. So the major contributor here is going to be going to our septum transversum. So let's move on to our next. So after formation of the neural tube, there is specialization of the intraembryonic mesoderm into 3 distinct regions. Paraxial mesoderm, closest to the notochord, develops paired clusters of cells along the length of the notochord, called somites. The sclerotome of each somite develops into what structure or structures?
Okay, final answers in 5, 4, 3, 2, 1.
All right, so looks like the majority of people chose B, and that is the correct answer. So remember our somites, that's the molecular clock of embryology. These are really cool structures. They form at a very distinct time frame in every species. Going back to my work with zebrafish, if I could count the number of somites, I knew exactly how old that embryo was. And that somite then gets differentiates into 3 kind of portions. We have our dermatomes, myotomes, and sclerotomes. So use the letter to help you figure out what it's going to be. So dermatomes is our dermis, right? So that's D. Myotomes, muscles of the body, so M. And then sclerotomes, we've got our spine and the muscles around that spine. So kind of use that trick to help you remember what those structures are and which part of that somite they're derived from. So well done with this. All right, our next one. Based on the rotational direction of the embryonic limbs, the flexors of the anterior upper limb are located in blank compartment, and the flexors of the lower limb are located in blank compartment. So try to match those 2 blanks with those answer options.
All right, we will close this poll in 5, 4, 3, 2, 1.
So let's see how we did on this one. So B is that correct answer. So our upper limbs, the flexors are located in the anterior portion area, and then the flexors of the lower limbs are located in the posterior. And so that's kind of dictated or pictured here in this image where you can see kind of the differences between the location of flexors and between the upper and lower limbs and which compartment they're located in. All right, so next question. So early in week 4, post-fertilization, neural crest cells begin to migrate into the primitive heart tube. During developmental weeks 6 through 7, these cells will separate the truncus anteriosus in the pulmonary trunk and the ascending aorta. The separating structure is known as which of these following?
Okay, get your final answers in 5, 4, 3, 2, 1.
This one's probably a little bit difficult. So the correct answer is going to be our spiral septum. Another name for this is our aorticopulmonary septum. So again, we need to separate these structures during development. And you can see kind of in that final image here, we have our aorta and then we have our pulmonary vasculature there, and that's going to get separated by that spiral septum during embryology. So our endocardial cushions, that's going to separate, help create our valves and kind of make our septum. And it's the same thing with the interventricular septum. That's going to help kind of divide our ventricles apart. So spiral septums is our best choice. All right, so let's move on to our next question. So oxygenated blood that leaves the placenta travels in the umbilical vein into the umbilical cord. Some of this blood is directed into the liver, but much of it is shunted around the liver to enter the inferior vena cava. What is the name of this shunting vessel?
Okay, polls close in 5, 4, 3, 2, 1.
All right, so the most people chose C, and that is correct. So that's our ductus venosus. So you can see here this image, so our umbilical vein, and that shunt there is going to be that ductus venosus on its way to our vena cava. So well done with that one. All right, so next question. Several hours after birth, a full-term neonate is observed to have considerable drool and white frothy bubbles at the mouth. The infant experiences a brief episode of coughing, followed by gagging, choking, and cyanosis, so they turn blue. The most likely diagnosis for this baby is.
Okay, polls for this question will close in 5, 4, 3, 2, 1.
Excellent. So our correct answer is C, and most of the people chose that one. So well done. So all of these sort of symptoms are associated with that tracheoesophageal fistula. And this is where we don't get proper separation of the esophagus and trachea. And that's going to cause some of these. So inadequate surfactant production is not necessarily going to come along with these same types of symptoms, especially things like drool. That's not going to come out from that type of presentation. All right, excellent. So let's move on to our next. So the development of all the bronchi, primary, secondary, and tertiary, occurs during which phase of lung development?
Just want to say again, you guys are doing so great with participation. This poll will close in 5, 4, 3, 2, 1.
All right, so let's reveal our answer. So the correct answer is embryonic. So I think the key here is just understanding what the bronchi are. So that is that first portion of our trachea. And and the entire respiratory tree, and it develops just like that. It's a tree and it has multiple generations. So if you think of every branching event as a generation, there's over 20 generations or so within our total tracheal tree. The latest portion of that are going to be our alveoli and our acinus of those. And you can see here the different time frames of each development of each structure. So the bronchi are essentially our trunks of our tree before we start branching off. And so they're going to be occurring first. So that's going to all be during our embryonic. So that's how you can kind of differentiate the different time frame when these various structures of our tracheal tree are going to develop. So our bronchi are basically our trunk and the first couple of branches that come off, and that's going to come first. So that's during our embryonic period. Excellent. So let's move on to our next. So the jejunum, ileum, and cecum and the ascending colon receive oxygenated blood from a major artery of the abdominal aorta. Identify this blood vessel.
All right, polls for this question will close in 5, 4, 3, 2, 1.
Excellent. Okay, so we've got the majority of people have chosen B, and that would be our correct answer. So these different parts of our GI tract are going to be fed by our superior mesenteric artery. Kind of see that explanation here in this image of, so renal arteries, that's going to be our kidneys, and those other ones also don't really feed these structures. So well done with this. So again, just kind of review where these blood vessels will go to and what structures they'll provide oxygenated blood with. Excellent. All right, here's one near and dear to my heart. So it's a kidney. So the ureteric bud from the mesonephric duct influences the formation of the metanephric blastema. What renal structures develop from the ureteric bud?
All right, this poll closes in 5, 4, 3, 2, 1.
All right, well done with your attempts on these. This is a tough one. So minor and major calyces. That is, so there's two structures or two cell types that give rise to our kidney. One is our metanephric mesenchyme or our blastema. So that's a mesenchyme cell type, or the epithelium from our ureteric bud. And so we've got 4 answer choices. The loops of Henle, the glomerulus, and our proximal and distal tubules, those all come from the metanephric blastema. The rest of the kidney, which is pretty much just our collecting ducts and our minor and major calyces, these are going to be derived from our ureteric bud epithelium. So that's kind of the differentiation within the metanephric kidney of what structures come from which origin. So most of our epithelium comes from the metanephric blastema, while collecting duct system and the minor major calyces come from that ureteric bud epithelium. So just differentiating between those two types of tissues and their derivatives. So that was a tough one, but well done on that one, and I appreciate all of the answers. So let's move on to this next. So the fetal portion of the placenta is derived from cells of the chorion that form long, thin, tentacle-like extensions that extend into the pregnant endometrial layer of the maternal uterus. What are these chorion-derived extensions called?
Polls close for this question in 5, 4, 3, 2, 1.
Great, so well done on this one. So the correct answer here is villi. So these are like finger-like projections that help that implanted embryo to kind of dig deep into the endometrial layer and gain access to blood and other nutrients from the maternal sources. Microvilli is kind of a trick one. These are small cell-like extensions on epithelial cells, but not necessarily the structure that we're talking about here. And then we've got two different areas of our decidual layer, our decidua basalis and our decidua capsularis. These are not involved in kind of digging deeper into the endometrial layer. That's going to be our villi. So that one's another tough one, but you did a great job on that one. Okay, so the facial muscles, their main actions are to facilitate facial expressions like smiling and frowning. These muscles are derived from the pharyngeal arch 2. The cranial nerve that innervates all these muscles is also a derivative of pharyngeal arch number 2. What is this nerve?
All right, and everybody get your answers in for what will be our last question of the night. We'll close this poll in 5, 4, 3, 2, 1.
All right, so we've got a lot of people with answer B, and that would be correct. So well done. So again, just connecting your pharyngeal arches to which nerves, cranial nerves that they will create, and what do those cranial nerves innervate. And so we're dealing here with, as you can see through the image here, pharyngeal arch number 2, that's going to be our facial nerve. So excellent, well done, everyone. And I hope this was helpful to figure out what gaps you still have and also validate what you were able to learn.
Thank you so much, Dr. Camarata. And just as usual, thank you to all of our presenters for doing such a great job and compiling these questions and just making this another great, excellent interactive review session. So thank you, Doctors Werner, Macnow, I see still on here, and Camarata as well. So thank you, huge thank you to all of you. The only closing remarks that I have is just I want everyone that's traveling to the regional competition to have a very safe travel. We are so excited to see you and best of luck. We're so excited to see you there and just again, really proud of everything that all the time you've put in to studying and all the efforts you've put into this extra efforts in anatomical sciences. I think it will really serve you well moving forward.
And if I
Anything-
might, Dr. Proskura, I just wanted to, for all of you still here, embryology is hard, and we know that, and medical students think it's hard. So I think what this maybe points out is that you know the anatomy, you're working on the histology, but sometimes you have to kind of devote now the time that you have left to some of those topics that are really the hardest. So kind of prioritize your time with what you did, what was most challenging for you tonight. And the other thing I would ask you to consider, and faculty members rarely say this, but I'm going to say it, is that, and Dr. Camarata might be right there with me, is that if you want additional practice questions, AI can generate those for you. And so if you would put these learning objectives, especially the regional learning objectives, which are more difficult, more challenging, put those into an AI website. And there are some that are better for healthcare professions and medicine, and ask that you get second order questions. It would give you a whole bunch of questions to work through, and I think you would find that very, very helpful. So best of luck, everybody. We'll see you soon.
Thanks again, everybody. Have a good night.